This video is going to cover the anatomy of the eyeball. So first things first, the eye has three major layers. The first is known as the outer tunic. Sometimes it's also called the fibrous tunic. So the fibrous tunic is composed of two parts. The first in blue here is known as the sclera. And the other part is the cornea. And this right here is the cornea. So the sclera is the outer layer here in blue. It is like the white part of your eyeball when you see it in a person in real life. And the outer tunic is very strong and protective. And the sclera also acts as the insertion point for these extrinsic muscles which move the eye. And then the cornea, which is this front kind of bulging convex portion of the outer tunic, that is it has to do partly with vision, and it helps bend the light and focus what we see, which we can talk about later. The cornea is clear, and it can sense pain, and it is avascular, so there's no blood vessels in this portion here. On the outside of the sclera is this membrane here known as the conjunctiva. And the conjunctiva is a mucous membrane. It's used for protection. It lines the inside of our eye socket, like the membrane there. And it holds blood vessels and is used for protection. So next let's talk about the inner layer, which is known as the retina. Label that here. This is this portion in black here. And the retina we'll talk about in more detail later. It is composed of all the rods and cones and other neurons in your eye. So this is this thin, delicate layer here, and all these fibers, these nerve fibers, meet at the back and converge and form the optic nerve at the back of the eye. And that leaves the eye at the back there. So now we have the outer layer as well as the inner layer. This space here that is surrounded by the retina is known as the posterior cavity. And that is filled with vitreous fluid. So this whole space is a hollow opening filled with vitreous fluid, which is a viscous gel-like fluid. And that's what maintains the pressure inside the eye. So this fluid is pushing against, and that's what's holding the eye together and maintaining that pressure there. So it also, it helps maintain the shape of the eye. So let's talk about the middle layer, which I've drawn here in pink. It's between the outer tunic and the retina, and that's known as the choroid coat in pink here. So the choroid coat is between these layers, like I said, and once it passes the retina here, it forms these structures known as the ciliary bodies. So the ciliary bodies are muscles that can contract or relax to help shape the lens, which we'll talk about in just a second. So attaching these, the choroid coat also makes up the iris here, which is, if you looked at an eyeball of a person in real life, that's the colored portion right here. So this colored part is the iris here, and this is a muscle that extends down, and the space in between the iris muscles is called the pupil. So that's the black part of someone's eye that you see. That's the pupil. And how this actually works is, if we have, let me draw it here. So if we have someone's eye like this, and let's say that this opening right here is the pupil, I'll color it black just so it looks like how you'd see it in real life. So that's the pupil. Well, then this whole pink part I've drawn is the iris, and the iris is composed of two different types of muscles. It's composed of circulatory muscles, which go like this, and then radial muscles, which radiate out from the center. And these muscles work together to expand and contract to allow more or less light into the eye for when we're seeing. So say these muscles go inward, your pupil is going to get smaller, or if they go out, it's going to get larger. So that's the contraction or dilation of your pupil, like you've heard when you go to the eye doctor or something like that. If your circulatory muscles contract, your pupil is going to get smaller. So if these muscles contract, it's going to close in on the pupil like this. So if these circulatory muscles contract, it actually gets smaller, and the iris gets larger. But if these radial muscles contract, you can imagine them pulling this way, the pupil is going to get larger. So that's how those two types of muscles work in conjunction to change the shape of the iris and the pupil. So I'll just write that here. We have circulatory muscles, as well as radial muscles. And that's how you change the shape. And it lets light into more or less into the eye. So that's the iris. Now let's talk about the ciliary bodies. The ciliary bodies are also muscle. They are attached to the lens, which is here in orange. And they're attached to the lens by these suspensory ligaments. They're called suspensory ligaments because they suspend the lens. And just like how the muscles in the iris change the shape of the pupil, the suspensory ligaments and the ciliary bodies work to change the shape of the lens. So the lens is part of the eye that also refracts light in order to see. So when light comes in, it hits the cornea, is refracted, and then refracted a second time when it hits the lens. And that's what concentrates the light onto the back of your eye to see, which we can talk about later. So the lens itself is clear. It has no blood vessels, and it is many cells wrapped together. If you've ever dissected it, when you cut it in half, it kind of looks like an onion. It's many, many layers. And it's suspended by these suspensory ligaments by the ciliary bodies. So when you see light is brought in, and the shape of the lens will change in order to refract the light differently. So if the lens is flat, that means you're seeing something from far away. So let me just draw a little cross section here. So say we took this part and cut it here, something like this. And this one will be for far vision, and this one will be for near vision. So here's our lens right here. And when you're seeing far away, your ciliary muscles are going to be relaxed, and the suspensory ligaments are going to be tight. So we're going to draw them like straight like this. And because these ligaments are pulling on them tightly, the lens is going to be flat. So you can imagine it more like this kind of shape, because it's being pulled on tightly. And when you see far, and so that allows the light to be bent in a different way to help you see farther away. And when you're looking at something close up, the ciliary muscles are going to contract. That's going to cause these to relax, the suspensory ligaments to relax. So let me draw it here. They're kind of relaxed. See, they're not as tight. They have slack to them. And that allows the lens to be less flat, less tight, less pressure on it, and it kind of bulges out more. It's more convex. So that's how the shape of the lens would change with different vision, far or near. And that's how the ciliary bodies and the suspensory ligaments work to make that happen. So so the last thing we have to talk about is part of the cornea, which I forgot to mention, called the anterior cavity. So we have the posterior cavity in this main part of the eye. This section where the cornea forms, we have the anterior cavity. And the anterior cavity is also filled with fluid, just like the posterior cavity, but this is called aqueous humor. And the aqueous humor is produced by the ciliary bodies, and it drains through the canal of Schlemm out of the eye. So that's pretty much all you need to know about the eye anatomy. There's three main layers: the fibrous or outer tunic, composed of the sclera and the cornea. Then we have the inner tunic, the retina, which bundles at the back to form the outer, the optic nerve. These two compose the two cavities of the eye, the posterior and anterior cavity, respectively. Then we have the middle layer, known as the choroid coat, which forms the ciliary bodies and the iris muscles. As we know, the iris forms the pupil, the space in between the iris muscles, and how they work in conjunction to change the shape, as well as the ciliary bodies helping form the shape of the lens for vision. So next we're going to talk more about the retina and the structure of that. So here I've drawn a close-up of the retina, the layers of the retina. So this part right here, this whole thing is the retina. So that means that this top layer is the cornea. In purple there. And this pink part is the choroid coat. And I guess when you think about it, the cornea is part of the outer tunic, which is the outer layer. Then we have the choroid coat, then the retina. I guess that this could also be the sclera. I'm not 100% positive though, but this is the outer tunic, the middle layer is the choroid coat, and then you have the retina, which is the inner lining of the eye, the inner layer. So this is not drawn to scale. This is just to show you there's two layers on top of it. The retina is actually very thin compared to the other layers, so just keep in mind that this isn't drawn to scale or anything like that. Okay, so the first layer of the retina is this pigmented epithelium. Which is comprised of melanocytes, which are special pigment cells. So this is this orange. They have the little dots on them represent the pigment and the melanocytes. After that first layer of epithelium, we have the next layer, which is the rods and cones. So these black structures here are rods. And the colored ones here, I've drawn one in green and one in red. You could also have a blue one. That is the cone. So a little bit about rods and cones. Rods and cones are known as photoreceptors. So they respond to stimuli by breaking down photopigments, which they contain within them. So photopigments are special pigments that break down when exposed to light to a certain degree. And there are all sorts of different photopigments. Rods have a specific photopigment called rhodopsin. And rods have rhodopsin in them. Rhodopsin is a certain kind of pigment that breaks down when exposed to light, and it is responsible for seeing black and white vision, and it can respond to like varying levels of light. So it can see in bright light, but it can also see in very dim light, and we'll talk about why in just a minute. But you need a very low level of light in order to use your rods. Cones, on the other hand, have photopigments that respond to green, blue, and red light, and they are all different. So that's why I've drawn a green cone here and a red cone here. You can also have a blue one. So this is red, blue, green, light responding photopigments. We're not going to worry about their specific names right now. So cones are different than rods because they respond to specific wavelengths of light, red, blue, or green, and they allow you to see color, whereas rods only let you see black and white. And you need a stronger stimulus. Well, not a stronger stimulus. They respond differently than rods, and I'll talk about that in a minute. But rods are they are highly sensitive but less precise, and cones are less sensitive but more precise. And like I said, once we get down to this layer here, we're going to talk about that. So the first layer of the retina is the pigmented epithelium, and right underneath that we have the rods and cones, which are the photoreceptors. After that, we have these bipolar cells. So let me write that here. So these bipolar cells synapse with rods and cones. And this is another difference between rods and cones we're going to talk about now. So with rods, you could have one bipolar cell synapsing with several rods like this. See how we have one bipolar cell synapsing with 3 here, or 2, or several more. I've just drawn it like that for the sake of this video. So this is known as convergence. So I'll write it here really small. So when you have one bipolar cell with several rods, that's known as convergence. And what convergence does, it's responsible for rod's ability to be very sensitive, but also less precise. So when the eye is struck by light, like this, the reason it's more sensitive is because light hitting anywhere in this region could activate one bipolar cell. So it's not one-to-one. So you don't need to hit light specifically here to activate this bipolar cell. You could hit it here or here or here or anywhere in between to activate this bipolar cell. So that means it's highly sensitive because you have more chance of light hitting this area and activating this bipolar cell. But it means it's less precise because when you're recreating your image in your eye, when the image is recreating, it doesn't know if the response made by this bipolar cell was from here or here or here. So you're going to have less of a sharp image in rods. And this is also why a very low level of light can, you can see in very low level of light with rods, because any light coming into this area can be seen or be detected by this bipolar cell. Whereas in cones, you have no convergence, so you have one bipolar cell to one cone. So now you have the opposite effect, where you're less sensitive because light has to hit right here, specifically on this cone, for this to be activated. But that gives you a more precise image, because when you're recreating that image, you know exactly where this came from, and it gives you a very sharp image. So again, with cones, you're gonna have a sharper image, and with rods, you're gonna have a less sharp image, but you're able to see a greater degree of light. So you can see in dim light versus bright light. It takes more for a cone to be stimulated perhaps than a rod, because there's more chance for it to be stimulated here. So for this reason, the cones will be kind of concentrated in the very back of your eye. And this is because, say you're looking straight ahead, your vision is most clear and sharp right and ahead of you. And as you fade away into your peripheral vision on the sides of where you're looking straight ahead is less and less focused, and it's, you could say, blurrier, less sharp. This is because when you're looking straight ahead, the light that hits your eye directly is going straight into the back of your eye. And that's where you want all the cones to be located, to give you that sharp image, because there's going to be a better chance of them being struck in the very back of the eye. Then as you go around the edges of your eye, you're going to have more and more rods, which gives you less acuity, but better light recognition, I guess. So that's the difference between rods and cones and how they relate to the bipolar cells. So where you have convergence, where there's one bipolar cell with several rods, that takes place in rods, versus no convergence with cones. After the bipolar cells here, they synapse with ganglion cells, which are depicted here. These are neurons, and these ganglion cells, as I said in like the first part of the video, they converge to form the optic nerve, which is right here. And the optic nerve leaves and joins at the very back of the eye and then leaves the eye. So how the whole thing works and how everything's related is it's called phototransduction. And phototransduction is taking light energy and producing an electrical signal from it. So changing that energy from light to electrical. So how it works is a photon, or a particle of light, hits the eye, and as it hits the eye, it will strike a photopigment inside a photoreceptor cell. So, like I told you earlier, photopigments are special pigments that are broken down by light. So as the as the photons or light strikes the photopigments, I'm just going to write this here. As the light strikes the photopigments, it breaks them down, and this creates a graded potential within the rods and cones. This graded potential could be transferred to the bipolar cell at the synapse if it's strong enough, and then the bipolar cell would carry that electrical signal by means of another graded potential into the ganglion cells. And if this graded potential is strong enough to reach the ganglion cells, it will become an action potential and be carried by the optic nerve to the brain to be further processed. So let me just write it here. When you have a graded potential, that could be found in rods and cones, as well as the bipolar cells. And then the action potential is only carried out by the ganglion cells. So light hits the eye, it breaks down the photopigment inside the rods and cones. This creates a graded potential carried throughout the rods, cones, and bipolar cells into the ganglion, which produces an action potential, carries the signal to the brain. And then we see. So that was the anatomy of the eye, including a closer look at the retina. I hope that helped.